Today's webinar is very timely with the scare of Zika virus emerging as a potential problem in the U.S. Today's speaker, Charles Cantrell, is a natural product chemist who has worked on insect repellents as well as other natural product solutions for pest management for the last several years. He received his PhD at LSU in 1998, followed by a postdoctoral position at the National Cancer Institute. This was followed by stints with USDA and private industry. He's been a chemist at the USDA-ARS Natural Product Utilization Lab at the National Center for Natural Products Research since 2004. He's co-author of over 100 peer-reviewed publications, inventor on 2 patents, associate editor of the journal Pest Management Science, and has been president of the Phytochemical Society of North America. He now serves on the executive board of AGRO. Charles, I'll turn it over to you.
Okay, thank you very much, Steve, and thank everyone for their attention and attendance. And I also want to thank Laura for really working behind the scenes to set up some of the announcements and things, and the rest of the AGRO for the invitation to present my research. What I plan to do today is start by just giving some rationale of why we look at natural products in general, just to kind of justify our existence, and I'll move into the process that we use, which I think is a very important part of what I want to present today, is the process that we take and the approach we take towards finding new active ingredients, new compounds that can be developed into commercial insect repellents in this particular case. And then I'm going to give some examples of folk remedy studies. One of the more mature studies was the beautyberry plant, and then I'll talk about the jatropha and breadfruit plants, and then the most recent one we work with is sweetgrass. And this work really spans about 10 years of research on this particular topic, this particular project. And much of this work is, of course, funded by USDA-ARS, but we also receive funding from the Deployed Warfighter Protection Program, DWFP, and may have a little more to say about that later as well. I think everyone knows what ethnobotany is, but I just mentioned it to remind us that of what we're doing here. We're really looking for traditionally used plants by native peoples and trying to determine if we can find some understanding for whether or not those plants do work, in fact, as insect repellents, those that are used traditionally as insect repellents. And furthermore, can we develop those into commercial products that can be used as insect repellents, which is really what our ultimate goal would be, or find novel compounds that can be modified synthetically to produce more active constituents. And I also want to mention this: what is an insect repellent? Because the term really varies quite a bit, and we're really not using it correctly, even as we begin to talk about some of the bioassays a little later in the presentation. In general terms, it's anything that repels or drives back an insect. But back in the 60s, I believe it's Diether published some more specific terms, which is any stimulus which elicits an avoiding reaction. And of course, you can break this down into both contact repellents and vapor repellents or spatial repellents. And but we're not going to get too deep into this. We're just going to use the term loosely, insect repellents. Many of the bioassays we're using are really biting deterrent bioassays. They're really not measuring spatial repellency or actual avoidance. They're measuring the number of bites that the insect makes. So technically they're more biting deterrents, but the term repellent is used synonymously in this case with that. And I'll also add that we're not just limiting ourselves to insects, we're also including more arthropods as well in some of the repellent studies that I'm going to mention, even though we say insect repellents. So let's give some rationale for why we look at natural products in general. I think no one would argue that they're typically more environmentally benign. They typically have co-evolved with other organisms, and they biodegrade very quickly. And they don't stay around very long like many of the halogenated synthetics, conventional pesticides that we may see. They're certainly a source of new molecular target sites, and this has been proven over and over. Nature has done a great job of screening through some of these possible molecular target sites available. Source of new chemistry, I mean, you really can't invent this stuff in the lab very easily synthetically, and you start to include the microorganism natural products and the marine natural products, and the chemistry is really is limitless as far as combinations that can be made, and nature has been making these modifications forever to control the insect infestation of itself. I'm going to have more to say about better public acceptance a little later, but I think we all know that there's a move afoot to in the organic pesticide, organic agriculture markets for more natural product-based alternatives for controlling pests in organic agriculture, and so we're seeing the public demand some of this a little bit more. There's certainly an evolved activity along with the organisms that are so there's been some fine-tuning in nature to perfect these natural compounds. Improved technology has made discovery simpler. I think this is given, and there's lots of examples of that with the NMR and database technology has been huge with preventing dereplication of a lot of the discoveries we're trying to make. And I'll have more to say about that as well. And of course, it meshes with biotechnology. We're going to see this even more so as we start to discover new natural compounds. Of course, the biosynthetic pathways are there, and the genes can be isolated and incorporated into crops, as we've seen with BT and other crops. In some cases, cheaper to register. I think in most cases, it's cheaper to register with the regulatory agencies. This came across my email just a few days ago, and I thought I would add it in because it sort of makes a point of the current demand, and USDA put this out as part of their census evaluation they do. And the number of domestic certified organic operations increased by almost 12% just in the last year, and this is the highest growth rate since 2008. So the point I want to make here is that the organic sector is really growing rapidly, and it's not going away. And what that means is that we need to have more natural product-based pesticides, and the consumers are demanding this. And it's going to continue to grow, and it's currently a $75 billion industry right now, and we better pay attention to this and have alternatives that are natural in the pesticide marketplace. And this same can be true with the insect repellent marketplace, and I got an example of that in a second as well. So some examples of pesticides that have been developed based on natural compounds, certainly your pyrethroids from the chrysanthemum flower, and you can, of course, most of them are discovered naturally, the pyrethrum extracts, and they're modified synthetically to produce some of your commercially available conventional pesticides, the permethrin, cypermethrin, deltamethrin, and other things. An example of a fungicide would be your strobilurin fungicides from the mushroom, and ultimately end up as conventional pesticides with your oxystrobins. And then the good example of herbicides would be your trichetones from the manuka tree, and there's some argument here as to whether or not they arose from the natural compounds, leptospermone or not, but needless to say, they're similar in structure, and they may have led to your mesotrione, your sulcatrione herbicides. Another really important example of a good example of the insects is the Dow product, the spinosades that are currently available, and this is, I'll have a little more to say about this later, but this is a good example of an insecticide that has spanned both the conventional marketplace as well as the organic marketplace with its OMRI approval, and it's basically shown us that yes, we can still discover new compounds from natural sources to work as insecticides and compete in the conventional pesticide marketplace. If you look at global sales of insecticides, you can see that the two of the big classes are pyrethroids, which I mentioned are derived from natural products. Your neonicotinoids are, of course, also originated from natural compounds, nicotine, and then your intact natural products. And altogether, these account for about 40% of the marketplace for global insecticide sales. So there's really a lot of rationale for why we should continue to pursue natural compounds as insecticides and insect repellents, and we're going to continue in this route. I only add this slide to mention if we're going to have a great impact on the agrochemical marketplace with natural compounds, you really would want to focus on certainly on herbicides, insecticides and fungicides, but herbicides really would have the greatest impact, and this is proven to be very difficult to confine herbicides that can be used in the conventional marketplace that are also natural. But nonetheless, this is really there's a lot of movement in this area, and people are trying to find something to be used competitively. Because I will throw these terms back and forth, conventional and biopesticides, I want to quickly define what I mean by this. It's really the EPA registrations, designations. When I say conventional, these are the most familiar you typically deal with. Mesotrione, picaridin, your deltamethrin, cypermethrin, all these names you're very familiar with, neonicotinoids, your pyrethroids all fall into this class. And then, of course, the biopesticides are your more naturally occurring substances as designated by EPA, and your microorganisms that control pests, your plant-incorporated protectants, things like carvol, neem oil, pheromones. And a lot of these you're seeing a lot more development in this area, this biopesticide registrations, and this is going to continue as well. We're not going to talk about antimicrobials. That's really more surface disinfectants, so I won't use that term at all. A review article we published just a few years ago looked at the numbers of products being registered by the Environmental Protection Agency, and we looked at those that are being registered as both conventional pesticides and biopesticides. And if you're just looking at the numbers registered and where their origins come from, about 36% of those are intact natural products being registered with the EPA. So there's a huge movement to get new natural compounds registered. Most of these are being registered as biopesticides. A lot of oil products, a lot of essential oil-based products that are being pushed into the marketplace because the consumers are demanding these products. Mint oils, orange oils, all these different oils that you can imagine are being produced into products and sold by many companies, even in places like Home Depot, some of the EcoSmart products and other things like that. Of course, synthetics account for about 31% of the numbers registered, and then you have synthetic natural derives about 5%, and then biologicals, which essentially are natural as well, are about 27%. Altogether, if you look at these other than synthetic, you're talking about 65% of the products being registered with EPA show their origins in natural processes in some manner, and so it's pretty significant. I'm going to mention this term crude extract throughout the presentation, and so I want to make sure I define it a little better. Most of you that are chemists probably understand a crude extract is just if you take something like coffee beans and you extract it with water, you're going to be remove the water, you're left with dried coffee, and this is really a crude extract of the coffee beans. And this term is used loosely by a natural products chemist. Of course, we can use various solvents, various organic solvents, and produce crude extracts, and we can do steam distillation and produce essential oil extracts. But all of these we kind of lump into this term crude extract to include anything from hexane to methylene chloride to essential oils to water extracts. And these are the ways that we use to extract the chemicals from plants, and each of these ways has their own distinct properties for pulling different chemicals from the plants, and we use those to try to find the constituents that are present in those organisms, those plants. So we typically try to source extracts and then screen them against different plant diseases, and we do this in a few different ways. We certainly can use random screening from plant repositories. What we're going to talk about today is really the folk remedy route, but I just want to remind us that we can also use chemical cues and target particular plant families. But ultimately, we can test any of those extracts from those plants against different diseases, and we do a lot of this in our research unit in Oxford, Mississippi, our natural products unit. We have programs for all of these diseases, but the one we're focusing on today is the insect repellents from folk remedies. But the pathway I'm showing in the next few slides is really the same. And the pathway we use is what we call bioassay-directed fractionation. And ultimately, our goal in this process is to find single chemical entities or single compounds that are responsible for the activity of the extracts as a whole. This doesn't always work out for us. Sometimes we lose activity, sometimes it's a combination of chemicals, but ideally we would like to end up with a single chemical entity as the commercial product or as a potential active ingredient in insecticide discovery and development. So the process, essentially, you grind the plant, you extract the plant, you produce different extracts, which we've already mentioned, and then you proceed to bioassay. And this is the important part, is that at each step of the way, we're going to rebioassay our materials. And in this case, it's an insect repellent bioassay or a biting deterrent bioassay. And I'll show you exactly what that assay is about in the next slide. But you proceed with purification using silica gel chromatography or liquid-liquid partitioning, and you rebioassay. So each step, I want to reinforce that we are proceeding back to the bioassay. You go to HPLC chromatography, you attempt to purify these compounds, these peaks in the chromatogram, you go back to bioassay. So ultimately we would like to get at a single compound or a set of compounds or a class of compounds that's responsible for the activity. And then we proceed with structure elucidation. And when I mentioned earlier that technology has really made this job easier, a lot of this is due, of course, to NMR technology, but I'm a real big fan of databases. So if you can do proton NMR and C13 NMR and do some high-res mass spec, maybe GCMS, LCMS, and then lead you to high-res mass spec, you can generally get to a molecular formula. And this is a key part when you have a compound you purify. You can put this molecular formula straight to databases, and you can save yourself a lot of time with structure elucidation. And this is typically the process we use, typically the purification process, and this is the structure elucidation process. Of course, if you're dealing with a new compound or something that's not showing up in databases, you have to proceed to the 2D NMR route and maybe chemical reactions, methylations, hydrolysis, acetylations to confirm the activity, confirm the structure of the compound. The 2D NMR techniques we typically use are nothing more than standard techniques these days. COSY, NOSY, ROSY, and HSQC and HMBC are typically enough for determining the structure of these molecules, and we're not going to go into any of that today. Let's move into mosquitoes now, and of course we all know that they transmit disease to 700 million people or more. I believe the death rate is currently 1 in every 17 people currently alive. The mosquitoes are responsible for death and causes as many as 3 million deaths annually. I believe this number has actually gone down, if I'm not mistaken. I did want to mention one thing here. We're going to move into repellents now. 38% of the U.S. population uses a DEET-based repellent every year. This is, to me, it's a remarkable number because I can't think of any other pure compound, single compound that is applied to your skin so generously as DEET is, other than something like detergents and soaps and maybe cosmetics. But some people, there were formulations of DEET 98% pure on the market at one time, and people are just smearing this on their skin, and it's a highly effective repellent, but I do think consumers are wanting more natural product-based repellents, and I'll give an argument for that as well. Of course, DEET is the most widely used insect repellent. Picaretin is approved in this country a few years back, is a good alternative to DEET. You've seen a lot more products with that. And another compound we're going to mention is picaretin, I just mentioned. Paramethane diol from oil of lemon eucalyptus is another one that is actually highly effective, at least according to the literature, as well as some of the common magazine literature, the Consumer Reports and other things. I show this slide only because I had an old version of this from Consumer Reports back in 2008, and this is the active ingredients column here. And what you noticed in that the old slide was that all of the active ingredients down here were natural compounds. So in other words, the least effective were your natural product-based repellents. We are seeing some of these move up the chain here now. I think the formulation is getting better, and of course you see a product like oil of lemon eucalyptus has a overall score of 87, at least by Consumer Reports, but I know the labs these were done in, and the data is very reliable. And so most of these are certainly picaretin-based, DEET-based, picaretin DEET, and most of your oils are down here. But I think this shows us that consumers are looking for alternatives. They're looking for more natural compounds that are going to be effective in this range up here. Most of these are effective at 7 or 8 hours against 2 species of mosquitoes and ticks. So this is the goal here, is to have things that are effective all day long that are natural product based. So the first example I want to give is an American beautyberry plant investigation we did about 10 years ago. This is the first one we initiated. It's probably the most mature project, and I want to explain exactly what happened with this one. But essentially it's the leaves of the American beautyberry plant. We were approached by a botanist that claimed that his grandfather would take cuttings of this leaf and just and cut a branch off and mash up the leaves and put it under the harness of an animal that was out plowing the fields, and it repelled the flies. This same botanist claimed that he used the leaves to rub on his skin when he was out in the field collecting plants, and it was a very good insect repellent. So we work with him, Charles Bryson, and other labs, Steve Duke, Jerry Clune, and Kamal Chauhan, and others that we worked through this project, and I'll mention some other labs as well that were instrumental in getting this compound discovered. So we set out to isolate what's in that. So to do this isolation, we have to have bioassays that can be miniaturized in some fashion, and this K&D module was one that really worked well for us. So we cannot go out into the field and apply an extract from a plant on skin in humans and test for repellency. There's lots of issues there that would prevent us from doing that. So we have to have really a model system. In this case, it's a biting deterrent bioassay that can act as our repellent guide or bioassay guide for fractionation. And this K&D module bioassay is proven to be very effective for using, and we're going to use this throughout all of these studies. We're going to use this same bioassay to get us started to find the chemicals in these traditional remedies. This bioassay, at one point it was using human blood of some sort. Now it's a modified version of that. But essentially you put a membrane on top of a well, and you treat a piece of cloth and place it over that well. So you have treatments, and you have positive controls and solvent controls. You separate them with a Teflon membrane, and you introduce the mosquitoes. There's mosquitoes in each one of these little individual chambers. You can do 6 at a time. You open a trap door, and you measure whether or not the mosquitoes bite through this collagen membrane, and whether or not they draw blood in. And you can measure this at the end of the 3-minute period. So it's a very short bioassay. Requires very little material, very little plant extract or pure compound, and you can from that you can arrive at a biting deterrency, or a ratio of bites to no bites, and give you an estimate of how effective your compound is going to be at preventing insect bites. Again, this term we use loosely, repellency, but we can say repellency, but it's really a biting deterrency. So if we took extracts from this American beautyberry plant, and what you see over here is a steam distillation. You see some yellow oil at the top. This is a typical steam distillation that produces many oils that you use every day, like mint oils and orange oils and other oils, but no different. It's the same process ultimately. It's really a hydrodistillation, not a steam distillation, but it's essentially the same thing. American beautyberry, if you were to look at the American beautyberry oil and compared to solvent control, and at a concentration of 100 micrograms per centimeter squared, what you would see is that the proportion not biting is very high here, which means it's acting very good at preventing insect bites. So that we want to understand this chart very well, because we're going to use it throughout the presentation. SS220 over here is nothing more than a DEET analog that was developed by USDA. I'd never made it commercially, but we use this in the bioassays as a positive control. DEET would give you about the same value. This is at a very low concentration, 25 nanomoles per centimeter squared, whereas our extract is still at a relatively high concentration. But again, we started by trying to choose a solvent that these other solvents, hexane, methylene chloride, we did many others, but these that you can see here is that the essential oil produced the active constituents. We proceeded with chromatography, silica gel chromatography, a few steps and iterations of that to arrive at the pure compounds in each of these fractions. And what I want you to see here is that fraction B in particular, the concentration is the lowest as per this axis, this y-axis here, but yet the activity is the highest. So one of the most dilute compounds in here had the most active activity of these fractions. We had other active fractions, compounds like intermediol, but this really was the subject of lots more investigation by our group, calicarbonal. And we really were able to show that this compound with intermediol and maybe humulene epoxide, some of these others, but primarily intermediol and calicarbonal were responsible for the activity in this folk remedy and works very effectively as an insect repellent. Calicarbonal was really a very unusual compound. It's a tetranorclaridane. They're much less common in the terpenoid literature because it's a truncated diterpenoid, and the compound has 4 stereogenic centers, as you see, and we set out to, it was never before discovered from nature, whereas intermediol was much widely known. But we set out to look at really both of these compounds, but we ultimately focused on calicarbonal and filed an invention disclosure and then a patent application on this compound, and tried to proceed with developing it as a commercial insect repellent. And let's go through some of that process here. So what is the activity of these compounds? Humulene epoxide, when you're testing it side by side with its DEET analog, it's not very active, but compounds calicarbonate and intermediol are both very active. They're equivalent to SS220, and this is testing against 2 different species of mosquitoes, an Aedes aegypti and a Anopheles stevensii, the malaria and yellow fever vectors. How does this compare with DEET? The activity was really slightly more active in the K&D bioassay, and you'll see a difference in when we get to another bioassay shortly. But calicarbonal was certainly significant above the control, had good repellency. Again, this is a short-term bioassay, but the concentrations are now very low even for our compound. They're equivalent to DEET, 25 nanomoles per centimeter squared, but yet they're performing as well as something like DEET. And it has a very pleasant aroma, I'll add. We proceeded to one of the next stages for repellent investigation. We collaborate with the guys in Gainesville, Florida, Uli Bernier's lab, and he has a cloth patch bioassay. And again, this is a you're protecting your skin here. You're not exposing the chemical, the calicarbonal, to your skin directly. You're protecting it with a membrane, but you're treating the cloth with the compound of interest. This would be an example of a blank where you're getting lots of bites on the skin. So how does this compound compare? We're looking for a minimum effective dose here. So the lower the number, the better. So you have a compound like DEET versus calicarbonal and intermediol. So what we noticed was that in this particular bioassay, again, this is a very low concentration. Calicarbonal was not as effective as DEET, but nonetheless it was very effective. We still had a lot of reasons to move forward with this compound. Tick repellency. So the next step would be to evaluate your compound against some tick bioassays. A lot of this was done in John Carroll's lab. We tested the black-legged tick and the Lone Star tick, the vectors for Lyme disease and ehrlichiosis in cattle. And the bioassay is very straightforward, very simple. Again, this is a miniaturized bioassay, which is very good for us natural products chemists, which have low amounts of compounds. Basically, you treat you protect your skin with a piece of cloth, and there's actually 2 layers here. And the top layer is treated with a treatment, and you put your finger in a petri dish with ticks. If the ticks make it to this line, they have not been repelled. If they fall off beforehand, and they've been successfully repelled. So it's a very easy method to measure actual repellency, whether or not they move away or move away from I guess the human host in this case. This is a black-legged tick in that assay. So how does this compare to something like DEET? As you can see, on a concentration basis, calicarbonal is as effective as DEET, and intermediol is certainly as effective as well. SS220 was used as a control in this assay as well. So we have great activity at a very low concentration against ticks. The next step is to really, we want to have these repellents work on a long duration, for a long duration. Turns out that calicarpanol actually does very well at this, even out to 3 hours at a very, very low concentration of 155 nanomoles per centimeter squared. We're getting very good repellency, 100% repellency at 3 hours, and it doesn't drop off until about 4 hours. Intermediate started to drop off a little sooner. So again, we wanted to move forward with calicarpanol. We did do some structure activity work with calicarpanol to try to understand some of the components. We did some oxidations, reductions with the aldehyde functional group and esterifications. We did some modifications to the olefin functional group, and we never made it to substitutions, but we did do some preliminary work. Just look at some of the more important ones that we did, and we published this in Chemistry and Biodiversity. But if you reduce the aldehyde down to a hydroxyl, the alcohol versus calicarpanol, there's really no change in activity. And very similarly, when you oxidize the aldehyde to an acid, the activity is still, even though it looks like it's different, it's really statistically about the same as the calicarpanol itself, but very effective still. So this functional group does not need to be an aldehyde; it can be modified slightly. However, when we modify, in this one we're going the opposite direction. When we modify this olefin, reduce it down using hydrogen and palladium, the activity is completely lost. So clearly, this functional group is definitely needed to maintain the activity. And again, when you combine the two modifications, the olefin reduction and the reduction in the aldehyde, you get in a compound that's lost activity compared to calicarpanol. So we have lots of evidence, and these are just a few examples of modifications we've made. Another modification we made, which I thought was interesting, was we took the DEET functional group, which is the diethylamine functional group, and added it to calicarpanol. And we also took the piperidine ring in piperidine and added it to calicarpanol, just because we knew that particular functional group was not, it didn't need to be an aldehyde, so we were like, let's modify it that way. Turns out the activity is actually reduced significantly, so really lost activity compared to calicarpanol itself. But again, these are lots of things that we try to do to determine if we can develop a synthetic alternative based on what we've discovered. So we did modifications that the aldehyde have little effect, modification dramatically affect the repellency there, and other unknowns. We sent calicarpanol off to Wyeth Animal Health Products, and it turns out calicarpanol does not work very well as a blowfly or fly repellent, at least in the bioassay that it was tested against. But so it has more effectiveness against mosquitoes and ticks. We did a lot of work with scale-up. So the question becomes, if you're going to move this forward, can you produce this compound in large quantities to develop it as a commercial product? Basically, the best thing we were able to come up with is you're still only going to, even bulk isolation, you're only going to end up with about 0.02% in the leaves, and of that oil that you distill is about 3 to 20% of it is oil of calicarpanol. We did a lot of seasonal studies, a lot of semi-synthetic studies, and we even did a total synthetic studies, which I'll mention on the next slide, was done by a collaborator of ours, that Manuel Theodorakis at UC San Diego, and he actually was able to synthesize this compound in about 6 or 8 steps, fine-tuning it, and we work with commercial partner SC Johnson and try to move forward with this. I think there are a number of reasons why it didn't proceed any further. Cost of production and effectiveness over the long term, I'm not sure it was active enough after a few hours to warrant moving forward. But I can list lots of different assays and steps in this process, but ultimately we were at a stage where we really were ready to move on to another folk remedy. And with that, we'll move into to the next one. And the next two really, I'll call them breadfruit and jatropha, because they ultimately have the end point was the same class of compounds. Breadfruit is used in the Hawaiian Islands for its production of its fruit, used as a flour alternative by some of the people in those Oceania areas and the islands, the Hawaiian Islands, and other places in that part of the Pacific. Essentially, the locals would actually use what they call the inflorescence, the male inflorescence of breadfruit, and they would take them to their living quarters and they would actually burn them as you would incense. And this burning of this breadfruit flowers was thought to effectively control mosquitoes in the living quarters and other places. So we set out to investigate this plant with collaborators Max Jones and Susan Murch and others at the Breadfruit Institute in in Hawaii. And again, we start the process as the same. I won't go into much detail, but basically the essential oils had good activity again from this male inflorescence equivalent to DEET. We were able to trap the burning smoke in DCM and water, and I'll show you on a later slide what the results of that were, but I will add that the essential oils of this breadfruit had activity better than DEET, and what was interesting is that we had these crystallization from an essential oil, which is actually very unusual. It's the first time I'd seen it. But the crystals from the essential oil had less activity than DEET, or less activity than the oil itself. But we did do the structural elucidation of that, and it was found to be a very common compound, palmitic acid. So this was the first indication that we may be dealing with fatty acids in this particular plant. We did the purification as we normally do, and the end result is that through a series of fractionations over about a half of a year, we were able to come up with the fatty acids C10, C11, C12. Saturated fatty acids were responsible for the activity of breadfruit. Palmitic acid has much less activity than any of these, but notice that all of these three fatty acids are more effective. Again, this is against Aedes aegypti, more effective than DEET. And we did do a combination effect, but the combination was really no different than C11 itself. We did some other blends as well, but none of them proved to be more effective than the individual C11 fatty acids. A third investigation we did, I'm going to go through because it really leads us back to breadfruit, was jatropha oil. Jatropha oil is used in India as a fuel oil, and it's also used for repelling insects. They'll burn an oil lamp. This is a seed-pressed oil. This is a little different than we're used to, but the seed-pressed oil itself is what we looked at in this particular instance. We did a similar study where we were actually able to burn the oil and trap it and get a DCM condensate and an oil condensate from combustion of the oil in an oil lamp using just a typical lab setup. But ultimately we went back to, you look at the oil, you evaluate the oil, jatropha oil against, I'll go back to this chart, it's easier to see. At a very low concentration, 10 micrograms per centimeter squared, you're getting activity that's good but not great. It's not as good as DEET. Ultimately, the pattern continued. We're getting compounds that are not as active as DEET, but they're statistically significant above control, ethanol control. But again, they're fatty acids, and palmitic acids, fatty acids that we've seen in the breadfruit came up again in this folk remedy from another part of the world. I'll remind us that the seed-pressed oils typically exist as not intact free fatty acids, but they normally exist as triglycerides, and we really were curious as to find out whether or not it was the triglycerides having the repellency, or if they did have any repellency versus the free fatty acids. And as it turns out, this is actually their first report in the literature on triglycerides having repellency, and we were actually able to show that the triglycerides triolein and tristerin, the ones that contain oleic acid and stearic acid, have fairly good, and tripalmitin, palmitic acid, triglyceride, have significant repellency above the control. Again, they're not as active as DEET, but they were statistically significant, and they do have really good repellency. And this is an interesting study which leads you to lots of ideas, because these triglycerides exist on your skin naturally. And so it remains to be seen how we can use this knowledge to develop products and other treatments maybe. Many of you may be wondering, well, what happens to the DCM condensate when you burn the oil? We did look at that, and the DCM condensate does contain free fatty acids, so burning the oil does give off fatty acids into the air, and so we were able to prove this and show this, and ultimately we published this Jatropha study in the Journal of Ag and Food Chemistry, or I'm sorry, in the Journal of Medical Entomology, published breadfruit study in Ag and Food Chem, and both of these ultimately resulted in fatty acids, which led us to a more systematic study on fatty acids themselves. So we really extended this using the knowledge we had to try to understand what's the best fatty acid. Turns out when you test all the fatty acids, the best ones are the ones that are being produced by breadfruit. It's the traditional remedy that's used in Hawaii. So the C10, 11, 12, 13 have the best repellency, at least against Aedes aegypti. We extended this to a time study. Again, we want repellents to be effective over the long period of time. It turns out that C11 and C12 saturated fatty acids, as well as the monounsaturated fatty acids, maintain their effectiveness as well as something like DEET. Even out to 6, 12, 24 hours, you're still getting some statistically significant repellency. So they work very well as repellents, at least in this K&D module bioassay. On a concentration basis, actually it's slightly more effective than DEET. DEET is the bottom line, so C12 actually works a little better than DEET. Let's skip that slide. So we went back to our good colleague Uli Bernier and evaluated these repellents in his cloth patch bioassay, and we were able to show that, again, it's he tested against 3 different species of mosquitoes, 2 Anopheles species and Aedes aegypti, and again we're looking at the lower number here as being better, the minimum effective dose. So compared to DEET, undecanoic acid, which is C11, performed very well, not quite as active as DEET, but very well at a very low concentration. And C12 was not as active as C11, at least in this bioassay. So C11 seems to be the focus of a lot of our efforts these days in trying to see where we can proceed with this. We did not have any intellectual property associated with C11 or C12 repellents intact natural products, because the literature is, if you go way back, you could see that there is evidence that these fatty acids do have repellency against mosquitoes. So this is not new information, but what we were able to show is that the folk remedies do produce chemicals that are effective insect repellents, and we've been able to show this with many of these studies that we've done so far. The last study I'm going to go through is the most recent study, which we presented at the ACS meeting a year ago, or less than a year ago, a sweetgrass investigation, and one of the colleagues that helped with the Breadfruit Institute from the Breadfruit Institute is now a professor at the University of Guelph, and he came to me with an idea for a traditional remedy that's actually used from sweetgrass, and sweetgrass grows in the northern United States and southern Canada, but he was approached, and it's used in a lot of other, like basket weaving and medicine, incense, perfume. But what we're more interested in is the fact that the flatheads of Montana would store really braids like you see below of this sweetgrass, and it imparts a sweet scent to their clothing, and they claimed that it prevented the insects from biting. And they would also burn. It's burning here. You can see on the right-hand side. They would burn the sweetgrass to keep the mosquitoes and biting insects away. So we did an investigation, as we were done for the last few. The flatheads of Montana and Blackfoot of Alberta Blackfeet also use this folk remedy. So cutting right to the active ingredients, you're gonna see really 2 compounds here that really come out, and that's coumarin and phytol. Again, they're as active as DEET, but not quite as good as some of the fatty acids we've seen or carbonyl. But nonetheless, they were associated with the activity. We did the bioassay guided purification technique and successfully found the active constituents through the same process we've used before. Coumarin is actually an interesting compound because it's not currently registered as a pesticide, but many of you may recall that the Avon Skinsosoft was really rumored to be effective as an insect repellent, and this was back in the 90s, I believe. It turns out a scientist did a investigation of the chemicals that Avon was putting in Skinsosoft, and they came up with the fact that it is coumarin in the Skinsosoft that's responsible for the repellency. So it's interesting that we go from a folk remedy like sweetgrass, or a traditional remedy, to something that is already on the marketplace that is well known to have some type of insect repellency. So we validated this folk remedy usage. And it's very possible that this led to Avon launching a bug-guard line of products containing picaridin, but I'm not sure that they're not based on coumarin, because coumarin is not registered as a repellent with the EPA. Okay, to summarize a few things that I just wanted to reaffirm is that the approach that we're using and the process has been proven in really 4 cases that I've shown you. So we can go from an idea of a traditional remedy being used to an extract, to a fraction, to ultimately a pure compound, and show that it is effective and that this process actually works, and we're able to use this to try to move forward with developing these as commercial products. So the traditional use has been validated. And I mention others here because I only mention the ones that work. We have others that people come to us with, and we do the extractions and we find out, well, there's no repellency here, so the project dies. So they don't always work, but in these cases they've proven to be very effective insect repellents in projects. And options for development, of course, you can look at all of these, and it's interesting to look at the breadfruit and Jatropha, because both of these products are burned traditionally. So it would make sense to look at those not only as topical repellents, but also as spatial repellents. And we're actually doing some of that right now and trying to see if there's a way to develop these fatty acids into commercial insect repellents and move forward with their development. And we're making some progress in that way. And let me go back. And I mentioned here perhaps traditional usage, because to me this is important when you have something like breadfruit, and there is a company that actually is trying to import the male inflorescence from breadfruit to be used in a traditional manner as a insect repellent, just as the natives of Hawaii or the native people of Hawaii would use it as burning it in their living quarters as a insect repellent. So there is a possibility of seeing this developed in a more traditional manner, other than just the pure active ingredients, which is another approach we're using. And lastly, it's thank you for your attention and your time, and I certainly want to thank funding sources USDA and DWFP, and I'll remind you to support your agrochemical division. If you're a member of ACS, please consider joining these divisions. This is where all the legwork gets done in organizing these webinars as well as these symposiums at your annual conferences. And I'll thank you again for your attention. I'll be happy to answer any questions. Thank you.